Swayam Prabha. Digital India. Educated India.
Hello learners, I am Dr. Rekha Sharma, former professor, UGC, Human Resource Development Center, Rashtrasant Tukroji Maharaj, Nagpur University, Nagpur. In this video, we shall study enzymatic browning. Now let us first study what is browning reaction. When fruit and vegetable tissues are injured in any way or peeled during processing, a darkening of tissues called the browning reaction occurs. Some browning reactions are enzymatic and occur in fresh living tissue or at least in tissues that still contain enzymes. When enzymes are denatured by heat or any other reaction, this reaction no longer occurs. Now, enzymatic browning occurs in many tissues whenever they are injured. The injury can be the result of bruising, cutting, freezing, or disease. That part of the injured fruit or vegetable which is exposed to air undergoes a rapid darkening. Now, let us see what are the effects of this enzymatic browning. Enzymatic browning is detrimental to quality maintenance of the fresh cut fruit and vegetable industry, although it is beneficial to the development of color and flavor in some food items such as tea, coffee, and cocoa. A variety of fruits and vegetables such as apple, pear, banana, peach, lettuce, and potato are especially susceptible to enzymatic browning during processing and storage. Browning not only has a negative effect on their appearance but also may impair their sensory properties, including taste, odor, and texture, as well as nutritional value. Now, enzymatic browning is a widespread color reaction occurring in fruits and vegetables, which involves the interaction of oxygen, phenolic compounds, and polyphenol oxidases called as PPOs. Enzymatic browning does not occur in intact plant cells since phenolic compounds in cell vacuoles are separated from the PPO that is present in the cytoplasm. So, the enzyme is in cytoplasm and the phenolic compounds are in the vacuole. Once tissues are damaged by slicing, cutting, or pulping, however, the mixture of PPO and phenolic compounds consequently results in a rapid browning reaction. Enzymic browning is caused by enzymes collectively referred to as phenolase, phenol oxidase, polyphenol oxidase, catecholase, chrysolase, and tyrosinase. These enzymes are widespread in microorganisms, plants, and animals, including humans, where its action leads to skin pigmentation. It contains copper in its active site, which is essential for enzyme activity. Now, let us study what happens in the enzymatic browning reaction. This polyphenol oxidase catalyzes two basic reactions. First, hydroxylation of the phenolic substrate at the ortho position, that is adjacent to an existing hydroxyl group, monophenol oxidase activity. And second, oxidation of diphenol to orthobenzoquinones, that is diphenol oxidase activity. This is monophenol. The oxidation occurs by polyphenol oxidase and it forms a hydroxylation of phenolic substrate at ortho position. This is at the ortho position and this diphenol is formed, and after this diphenol again oxidation occurs and this diphenol is converted into orthobenzoquinone or orthoquinone written here. Okay. Then both reactions use molecular oxygen as a co-substrate. Quinones undergo further non-enzymatic polymerization leading to the formation of pigments melanin. Now let us study more about these phenol oxidases. Phenol oxidases in fruit and vegetable tissues exhibit optima in the general range of pH 4.0 to 7.0 and some substrates influence the pH optimum. Temperature optima for phenol oxidases are in the range of 30 to 50 degrees Celsius, but temperature stability is comparatively high and characterized by half-lives of several minutes in the range of 55 to 80 degrees centigrade depending on source. Thus, during thermal processing, ample opportunity exists for phenol oxidases to become activated. Since temperatures approximately 60 to 65 degrees centigrade evoke cellular leakage, that is decompartmentation, and mixing of enzyme and substrate at elevated temperature. Now let us see what are these phenolic compounds or substrates. The phenolic compounds of fruits and vegetables are diverse and vary in accordance with species, cultivar, maturity, and other plant physiological conditions. Now, these are some of the polyphenol oxidase substrates: tyrosine, caffeic acid, catechol, p-cresol, pyrogalol, O-caffeol, and epicatechin. Structurally, they contain an aromatic ring bearing one or more hydroxyl groups together with a number of other substitutes. Phenolic compounds and PPO are in general directly responsible for enzymatic browning reactions in damaged fresh cut products during post harvest handling and processing. Now, how to prevent this enzymatic browning? The approach for the prevention of enzymatic browning is divided into physical and chemical methods. Physical methods to regulate enzymatic browning include thermal treatment, prevention of oxygen exposure, use of low temperature, and irradiation. Heat treatment, such as blanching, can easily inhibit enzymatic activity because enzymes which are composed of proteins are denatured. In the wine making process, enzymatic browning is suppressed by heat treatment at 60 degrees centigrade for 3 minutes before brewing. Dehydration, freezing, and thermal processing are effective as long as the time required to affect the process does not permit intolerable browning and textural changes related to quality retention. Other physical means include packaging in modified atmosphere for minimally processed foods or coating of tissue section with sugar syrups, especially for frozen products, or edible films to limit oxygen co-substrate availability. Now, most popular are chemical treatments based either on inhibiting or deactivating enzymes, complexing native substrates, or reducing quinones back to orthodiphenols and or conjugating quinones in a manner that prevents melanin formation. The most important factor that determine the rate of enzymatic browning of fruits and vegetables are the concentrations of both PPO and phenolic compounds present, pH, temperature, and oxygen availability of the tissues. All the techniques attempt to eliminate one or more of the essential components such as oxygen, enzyme, copper, or substrate from the browning reaction. Consequently, dips in anti-browning solutions, modified atmosphere packaging, that is MAP, and heat treatment and refrigeration storage are the most common methods used to help maintain the initial color of fresh cut fruits and vegetables. Now let us study more about dips in anti-browning solutions. The use of reducing compounds, including ascorbic acid, that is AA, and its derivatives, cysteine and glutathione, is most effective for controlling enzymatic browning, and calcium ascorbate is already commercially adopted by the fresh cut produce industry. Reducing compounds can play an important role in anti-browning either by reducing orthoquinones to colorless diphenols or by reacting irreversibly with orthoquinones to form stable colorless products. Sulfides were found to be very effective in controlling browning. However, the application of sulfides on fresh and fresh cut produce was banned by the United States FDA due to its deleterious health effects on the allergenic populations. Ascorbic acid and its derivatives have been used in many studies in concentrations ranging from 0.5 to 4% and have been leading generally recognized as safe, that is GRAS antioxidants for use on fresh cut products for the prevention of browning and other oxidative reactions. Generally, its inhibitory effect is also due to the reduction of the orthoquinones generated by the action of PPO enzymes back to phenolic substrates. In addition, ascorbic acid is also believed to have a gelating effect on the copper prosthetic group of PPO. Gelating agents such as sorbic acid, polycarboxylic acids like citric, malic, tartaric, oxalic, and succinic acids, polyphosphates like ATP and pyrophosphates, macromolecules like porphyrins, proteins, and ethylene diamine tetraacetic acid, that is EDTA, which can inactivate enzymes by binding to transition metals in the metal enzyme complex, have been used for a variety of food processing applications. PPO possesses copper at its active site, and removal of the copper by chelation inevitably renders PPO inactive. A typical combination of anti-browning agents for fresh cut products may consist of a chemical reducing agent, an acidulant, and chelating agent. Now, modified atmosphere packaging. The rapid growth of the packaged fresh cut produce industry has been enabled largely by the development of MAP technology. With MAP, the desired balance of oxygen and carbon dioxide is created through the control of oxygen and carbon dioxide transmission of the packaging film and the respiration rate of the produce. Studies have shown that low oxygen and elevated carbon dioxide atmospheres can slow down the browning reaction. Now, heat shock and refrigeration. Heat treatment is the most widely used method for stabilizing foods because of its capacity to kill microorganisms and inactivate enzymes. Enzymatic browning in canned or frozen fruits and vegetables may be effectively controlled by treatment with steam or hot water at temperatures ranging between 70 and 105 degrees centigrade. However, because of the damage caused to the living tissues and the requirement for fresh quality, these methods are not suitable for the prevention of browning in fresh cut fruits and vegetables. Mild heat treatments in the range of 45 to 60 degrees centigrade has been used to induce the formation of heat shock proteins, that is HSPs, which afford a variety of benefits to fresh and fresh cut produce, ranging from browning inhibition to protection against plant pathogens. Now, balancing browning inhibition and pathogen inactivation. Fresh produce may be handled numerous times as it travels from farm field to dinner table. Contamination with human pathogens can potentially occur at any one of many stages, including in the field by animal carrier or irrigation water, during harvesting, transportation, packaging, or distribution. Unlike intact fruits and vegetables, the physical and chemical barriers provided by the epidermis of fruits and vegetables are removed during preparation of produce for the fresh cut market. This practice creates opportunities for spoilage or human pathogenic microorganisms to directly contact the edible portion of produce, thus leading to more rapid product decay and the potential for pathogen contamination. Wounding during processing releases plant cellular fluids which provide nutritive media on which pathogens, if present, may survive and grow. Now let us summarize. Enzymatic browning occurs in many tissues whenever they are injured. The injury can be the result of bruising, cutting, freezing, or disease. Browning is usually initiated by the enzymatic oxidation of monophenols into orthodiphenyls and orthodiphenyls into quinones, which undergo further non-enzymatic polymerization, leading to the formation of pigments. Physical methods to regulate enzymatic browning include thermal treatment, prevention of oxygen exposure, use of low temperature, and irradiation. Dips in anti-browning solutions, modified atmosphere packaging, and heat treatment and refrigeration storage are the most common methods used to help maintain the initial color of fresh cut fruits and vegetables. These are the references. With this, we have come to an end on enzymatic browning. Thank you.